Here you go, Markus.
Okay. Yeah, thank you. Sorry for the difficulties with the PDF slides. Yeah, thank you for the invitation and the opportunity to give a talk in this wonderful location on my current research on cfDNA fragmentation. So we've heard about cfDNA this morning already. So that are DNA fragments that are circulating in the blood, and they can come either as part of normal cellular turnover or from various pathological processes, and in particular from cancer cells leading to circulating tumor DNA. And it has been investigated for a long time as it carries rich biological signals about various pathological states, and we have heard it can be used to infer the tissue of origin as well. There have been more than 1,000 clinical trials worldwide, and there are a few FDA-approved tests, such as liquid CDX. So why is that? And the main reason is that we get information in a less invasive manner than a tissue biopsy, with fewer complications, and the results are rapidly available within a few days or sometimes even hours, like the COVID-19 testing. Applications so far in development are prenatal genetic diagnosis, when the DNA of the fetus is circulating in the blood. Okay.
Online colleagues to be able to see it, and for some reason they cannot. Let's try this. Does anybody have it?
Yes.
Good. Thanks.
Sorry. Okay. Thank you. So, I can see the slides. So yeah, we have the prenatal for like genetic abnormalities in the embryo or fetus, for early cancer detection, and also for surveillance and monitoring it in a longitudinal way. So how is the data collected? In general, ctDNA makes up less than 10% of the cfDNA, even at late tumor stages, and in early stages it might be only 1% or less, but this doesn't invalidate the approach if you know which signal you're looking for. The data is collected via bioassays such as polymerase chain reaction or sequencing. Particularly valuable would be long-read sequencing data, such as the recent Oxford Nanopore platform. So the main question is, can we go beyond just looking at the cfDNA concentration, but actually make predictions based on the entire fragment length distribution or fragment profile, and understand this and make more robust predictions of cancer. So we're interested in the fragment length distribution, which means that for each length X up to a possible maximum L, which is at most the length of the DNA molecule, but usually much shorter, how many fragments are there of length X? That's what we're keeping track of. Conceptually, this is modeled with fragmentation processes studied by mathematicians, probabilists, and physical chemists for a long time. For instance, there's a great introductory textbook by Jean Bertrand from around 20 years ago, and they have used these fragmentation processes to study rock mining for mineral extraction, and they have derived several theoretical results for the stationary distribution of the fragment length. So conceptually, it is that a fragment is entering the blood, and then there may be some fragmentation or splitting events or some elimination event with the red cross. And this gives you a distribution of the fragment lengths, and in the long term, you can assume you're in an equilibrium state of that process. So biologically, there are three kinds of events: immigration event, or shedding of mitochondrial or genomic DNA. The circulating DNAs is cutting it into smaller pieces with array-depending on the body fluid, health conditions, like individual properties. And the exit mechanism also varies. There's nucleosome-bound cfDNA, which is more stable and thought to be mainly cleared by the liver, whereas naked single-stranded DNA thought to be cleared by the kidney in a length-dependent manner. Yeah, and in general, I thought that the cfDNA is concentration-dependent elimination and has a reported half-life of 50 minutes to 2 hours, so it's very fast, the turnover. What the model tries to quantify is various aspects, such as the rates or speeds at which these events occur, and whether or in which way the fragmentation is dependent on the length of the fragments, and to clarify the exit mechanism. From a biological point, it's not entirely clear yet which of the three exit mechanisms it would be. So what this means is that the model is keeping track of this histogram of counts, independence of time. And there are three kinds of events: fragmentation, immigration, and exit. So basically, we're drawing exponential waiting times for each kind of event, parameterized by this fragmentation speed CF. And for fragmentation event, we have this fragmentation power alpha, which determines if it is negative, such as 1 divided by root x, that longer fragments are cut down more slowly than shorter ones, whereas if it's positive, the shorter fragments are broken down faster. And this beta random variable determines the ratio at which the fragment is cut, which could be uniform or rather towards the edges. And the immigration, we assumed if it occurs, we draw a uniform fragment, but in theory you could vary that maybe to test other distributions of the migrating fragment length. And for the exit mechanism, there are three proposals. The easiest conceptually would be constant, so it would be ignorant of the fragment length, just any fragment would be drawn and removed. Maybe more natural biologically would be a power law, meaning that longer fragments are cleared more slowly, and power law with a fixed boundary, so they're only cleared up to a certain threshold BE, and beyond that, yeah, the exit rate is 0. So what we then arrive at is a continuous time Markov process that can model these biological phenomena and allows to simulate this based on simulating a lot of events, the fragment profile, and compare the simulations to empirical data and establish evidence for which is the correct exit mechanism. And in the long term, hopefully to use it for diagnostic purposes. So we simulated this, like fixing all the parameters and varying one of them. So if we look at the fragmentation power, and as we increase it, the tail of the distribution becomes much lighter, and where the distribution has a power law, the fragment length distribution has power law with a slope that we fit, and that's decreasing as we increase alpha. And if we look at the dependence of the exit power, where we only consider negative exponents, as we increase negativity, also the tail becomes somewhat weaker. So what we've got is a fast simulation engine. We implemented this with this R package RCPP, which allows a relatively quick simulation of synthetic data, which allows simulation-based inference or ABC inference in order to estimate the model parameters for a given empirical fragment length histogram. And then the current plan is to use those parameters estimated from the data, maybe along with residual summary statistics, as features for machine learning algorithms such as this extreme gradient boosting or support vector machines for classification between healthy and tumor cfDNA. So just the data that we have so far is too small to actually do that. But the conceptual idea is to bridge this mechanistic model, which is kind of gives something interpretable with the predictive performance of XGBoost or support vector machines. So what we need to make sure first, though, is that the parameter estimates that we get from the ABC inference are actually reliable if on synthetic data when we know the parameters, and then to integrate this with XGBoost and compared or validated with clinical data. Based on the data we have looked at, it seems like this polar with fixed boundary is the most realistic one. That's where we are. Thank you.
All right, we are open for questions.
Thank you. That was very interesting. My understanding is that these DNases cut at like specific site, and there's different types of DNases which can be activated in different tissues, and that can change with the cancer type, and that affects the fragmentomics. Is that something you thought about? And also, again, kind of thinking about the size distribution here, that means there's like certain sizes that are typically enriched. Is that something that your model is able to recapitulate?
So regarding the DNAs, we consider different parameter values for the A. We don't have we had like a sample a data set of like 13, so we cannot really say anything. It seemed like this uniform distribution was the most likely one, but if there are different DNAs acting differently, then it might be reasonable to incorporate it into the model. So that's thank you for that point. And your second question was.
My second question was around the sort of like unevenness in the size distribution that result from that, and that specific fragments are typically enriched. And what I saw in yours were kind of very kind of exponential looking like distributions.
Like the tail the tail?
Maybe we can talk offline as well.
Yeah.
Yeah.
So what the fragment length distribution was exponential, so that's why you get this straight line in the log plot. But yeah, let's continue offline then. Yeah.
Sounds great. Thank you.
Thank you.
Right. Any other quick questions? All right. Let's thank the speaker again.